Morning, students. Today we are going to discuss about the male and female reproductive system. This male and female reproductive system histology is very important practically, because to assess the cause for the infertility in male and to assess the infertility cause in females. For these two purposes, female and male reproductive system histology plays most important role in detecting the cause for the infertility. Because if there is a infertility in a couple, you have to assess both the partners, male and female. If you want to assess that what is the cause for this infertility, you are supposed to take the biopsies, that is a fine needle aspiration biopsy from the testes from the male and ovaries from the female, and sometimes you also need to take the biopsy from the endometrium, or sometimes you have to check the status of the uterine tubes, or in the male you have to assess the seminiferous tubules, whether there is a production of the spermatozoa is there or not. That is also very important. Or is there any obstruction to the passage of the spermatozoa in the male, or is there any obstruction to the passage of the ovum in the female? These things you have to observe under the microscope. For this purpose, you have to study the histology of the male and female reproductive system in detail. At the same time, the prostate gland is also very important gland in the male. That is benign or malignant hypertrophy of the prostate gland is common in the elderly male persons. That is especially after the age of 60 years or 65 years of age. There is chances for the enlargement of the prostate gland. If there is enlargement of the prostate gland, it may cause obstruction to the urinary flow, because already you know the gross anatomy, that is the prostatic part of the urethra, it passes through the prostate gland. That is why there are more chances for the obstruction to the urinary flow. Because of this reason, we have to study in detail about what are the different male reproductive structures and what is the histology of the different structures, and female reproductive system also same. What are the common female or internal, especially internal reproductive organs of the female, and what is the histology and what is its functional correlation. That is also very important in histology. If you want to study histology properly, why you are supposed to know about the histology? First, you have to understand that what is the function, the structure of any structure in our body or any organ in our body. It depends upon its function. That is why the functional correlation is also most important. Today we are going to discuss in detail about each and every part of the male and female reproductive system. Okay, you see here, this is the male reproductive system. Here, this is the testes. This is the testes, and this is the epididymis, and this is the vas deferens. Then it goes and it enters into the prostatic part of the urethra, and this is the prostate gland. Before joining with the prostate gland, it also receives a duct from the seminal vesicles. Seminal vesicles together they join as a ejaculate duct into the prostatic part of the urethra. Okay, this is the normal pathway of the passage of the spermatozoa from the site of production, that is at the testes, that is within the seminiferous tubules. These are all nothing but the seminiferous tubules. This is each one is the lobule, and this testis, it is covered by a outer layer, that is the tunica vaginalis. This layer is called tunica vaginalis. Again, it is having two layers. That is, outer one is the parietal layer, and inner one is the visceral layer. And inside this tunica vaginalis, again there is a white colored area. This layer is called tunica albigine. Tunica albigine. Albus means white. Albus means white. It is white layer. That is why it is called tunica albigine. And inside this tunica albigine, again it is lined by another layer. That is the tunica vasculosa. Tunica vasculosa. and within the tunica vasculosa, it is divided into many lobules, and they are filled with the seminiferous tubules. Seminiferous tubules. These are the actual site for the production of the spermatozoa. These are the actual site for the production of the spermatozoa. And here, this is the each seminiferous tubule is about 50 to 80 centimeters in length. So, to stretch the seminiferous tubule, it is about 50 to 80 centimeters in length. And within the each seminiferous tubule, there is a production of spermatozoa. And from here, these are reached to the mediastinum. This part is called mediastinum testis. Mediastinum testis to the red testis and different ductules. Then finally, together they form another duct. This is nothing but the epididymis. Epididymis. Then it is the tail of the epididymis. It continues as the vas deferens. And all these structures we are going to discuss. It is testis. What is the histology of the testis? What is the histology of the epididymis? Vas deferens, seminal vesicles, and finally prostate gland. These are all very, very important. Because of this reason, we are going to study. And all these structures, the structure, the microscopic structure of all these organs, it is functionally modified. Functionally modified. First, we see about the testes. Okay, testes. What are the most important and characteristic features of the testes? By seeing these features, we can say this is the testes. Okay, because we are having spotters in the histology exam. We are having spotters. Within 1 minute, you have to identify the spotter and you have to write the characteristic features. Okay, suppose if the slide is testes, what are the characteristic features of the testes? First, it contains seminiferous tubules. Seminiferous tubules. This is the very, very important because this is the actual site for the production of the spermatozoa and interstitial cells of Leydig. Interstitial cells of Leydig. And in between the seminiferous tubules, there are spaces. There are spaces. These spaces, they are filled with the cells. That is, these cells are also very, very important. That because this is the site for the production of the testosterone hormone. That is nothing but the male sex hormone. Male sex hormone. That is the testosterone. That is the site for the production of the testosterone is the Leydig cells. They are also called interstitial cells of Leydig. Interstitial cells of Leydig. That is the importance of the interstitial cells in between the seminiferous tubules. Next, within the seminiferous tubule, there is a spermatogenic cells that is at various stages. At various stages. You see here, or you see further magnification. You see. Okay, see these are all nothing but the each one seminiferous tubule. This is another seminiferous tubule. Like this, these are all the seminiferous tubules. And in between the suppose if you see this is another seminiferous tubule, this is another seminiferous tubule, this one. In between these are the cells. They are nothing but the Leydig cells. These are the cells responsible for the production of the male sex hormone. That is nothing but the testosterone. Testosterone. This is the male sex hormone. It is produced in the Leydig cells. Okay, these are all the seminiferous tubules. Okay, you see, if you see further magnification, if you take one seminiferous tubule, outside the seminiferous tubule there is a lining epithelium, and above this, outside this lining epithelium, there are mesothelial cells. These mesothelial cells are responsible for the squeezing the seminiferous tubules to expel the spermatozoa which are produced in the seminiferous tube. Okay, during ejaculation or during routine movement of the spermatozoa. Okay, here you see, if you take each seminiferous tubule, there are different types of cells. There are different stages of cells. Actually, there are different stages of cells. First and foremost important one is the supporting cell. They are called sustentacular cells or Sertoli cells. Sustentacular cells are Sertoli cells. Sustentacular cells are Sertoli cells. In between the sustentacular cells or Sertoli cells, there are spermatogonia and spermatocytes. Okay, there are different stages of the maturation of the spermatozoa. First, the supporting cells or sustentacular cells are Sertoli cells. These are the in between the two Sertoli cells. There is a tight junction. There is a tight junction between the two Sertoli cells. You see, this is the one Sertoli cell. This is another Sertoli cell. Here you see there is a tight junction between these two Sertoli cells. Here it separates the spermatogonia from the remaining stages of development of the spermatocytes. Okay, this is called blood testis barrier. Blood testis barrier. This blood testis barrier is produced by the adjacent sperm, that is stem cells, that is supporting cells. Okay, this is the very important because it prevents the antigen antibody reaction between the testis blood and the spermatogonia. That is why this blood testis barrier is very important. If there is any damage to the blood testis barrier, the person's own cells they treat as a foreign body, and they may cause there is a engulfment, there is a phagocytosis of all the spermatogonia, and the person may become sterile. Okay, there is no motile spermatozoa, there is no production of the spermatozoa. That is the one important point, that is blood testis barrier. Next, coming to the what are the different stages of the spermatozoa which are present in the each seminiferous tube from outside to inside. This is very important. When drawing the diagram, you must draw all these cells in order. That is also very important. The outermost cells we see here, these outermost cells, they are nothing but the spermatogonia. They divide by the process of mitosis, and they produce type A and type B cells. Then they enter into the next type of cell, that is nothing but the first are primary spermatocytes. Then these also go under that is a division, that is by the meiotic division. There is a reduction in the DNA and the chromosome number. Then finally they form the secondary spermatocytes. Then these secondary spermatocytes again they form the another type of cell, that is nothing but the spermatids. You see, these are the spermatids. They are rounded cells, spermatids. Okay, from the formation of the spermatogonia up to the spermatids, that is a one stage. Then the spermatids, then they divide and they metamorphose from spermatids into the spermatozoa, and this process is called spermiogenesis. The total process is called spermatogenesis. Here this only the metamorphosis of the spermatids into the spermatozoa, we call it as spermiogenesis. Okay, these are all the different stages of the spermatozoa. You must draw if they ask for the histology of the testes. That is the one point. Within the seminiferous tube. Then these spermatids finally they form the spermatozoa. Okay, here you see one seminiferous tubule, this is another seminiferous tubule. In between, this is the space. The space, interstitial space. Here, this space is occupied by the interstitial cells of male. These are the cells which are responsible for the production of the sperm. That is a male sex hormone. That is nothing but the testosterone. Okay, so if you want to draw the histology of the testes, first you have to draw the basement line, basal membrane. Then outside this there are myoepithelial cells. Then within the lumen of the seminiferous tubule, first you have to draw the sustentacular cells, and then you have to draw the different stages of the spermatozoa, maturing spermatozoa. Okay, this is the spermatid, and this is the spermatozoa. Okay, these are the stages you must draw when drawing the testis diagram in the histology. Okay, clear? That is very. Why we are supposed to know about the testis? That is very important, because in some individuals there is no production of the spermatozoa, that is called agiospermia. Or sometimes there is a production of the spermatozoa is there, but there is a less in number. Number is very less. Because of this reason, we call that condition, you call it as oligospermia. Agiospermia, oligospermia, these are the most common clinical condition. If you want to diagnose what is the exact cause for the sterility in male, you you have to go for the testicular biopsy. Testicular biopsy. If you want to take the testicular biopsy, then there is a different procedure. They give some local anesthesia and they take the piece of the testis. Okay, they observe the piece of the testis under the microscope, and it gives you some idea about the what is the status of the testis. Okay, that is why you must know about the testicular histology. That is the first point. Next, coming to the next, after the production of the spermatozoa in the testis, they are transported from the testis into the another structure that is nothing but the epididymis. That is nothing but the epididymis. The epididymis, you see, this is the epididymis. Okay, in epididymis, it is having three parts. That is the head of the epididymis, and this is the body of the epididymis, and then it continues as the tail. Tail continues as the vas deferens. Vas deferens. Okay, this is the vas deferens. Here, this is the tail of the epididymis. This is the body of the epididymis, and this is the head of the epididymis. Okay, this epididymis, it is nothing but a single tube. It is nothing but a single tube. It is folded on itself. It is folded on itself, and it forms like a bulb. Okay, then if you see the ductus epididymis lined by pseudo-stratified columnar epithelium with stereocilia. Pseudo-stratified columnar epithelium with stereocilia. These are the cilia, stereocilia. And here, the most important difference between the testis and the epididymis is because they may look like a seminiferous tubule. They may confuse because it is looking like a seminiferous tube. Because of this reason, you must be very careful while observing within the lumen. There is a more number of spermatozoa, free spermatozoa at the same time. And here, in this area, you have to observe there is a pseudo stratification, but there is no separate layers of cells. Separate layers of cells. That is a pseudo stratification. At the same time, there is a myoepithelial cells outside this tubule. Outside there is a myoepithelial cells. But here you have to differentiate. That is it is only pseudo stratification. This is not the different stages of the maturing spermatozoa. Okay, here this is the only pseudo stratification, and then it is having cilia. That is also especially the stereocilia. Stereocilia, and outside this it is lined by smooth muscle fibers. Smooth muscle fibers. You see, these are all the smooth muscle fibers surrounding each ductule or each ductus. There is a smooth muscle fibers. Then within the lumen there is a spermatozoa. Okay, why there is a smooth muscles? While ejaculation, it must contract, and it must push the semen along with the spermatozoa into the prostatic part of the urethra. To perform this function, it is lined by smooth muscle. It is lined by smooth muscle. Okay, this is the structure of the epididymis. This is the structure of the epididymis. Here, the lining epithelium is very, very important. The epididymis, that is pseudo-stratified columnar epithelium with the stereocilia. And smooth muscle fibers are also present surrounding the ducture. Each duct. Here, it is nothing but a single duct. It is folded on itself. If you take the section at one level, it may cross so many parts of the duct, same duct. That is why we are having just like the seminiferous tubule apparatus. Okay, that is why you must be very careful. At the same time, there is abundant amount of spermatozoa within the lumen. Abundant amount of spermatozoa within the lumen. Here in the testis, that the lumen may also contain the spermatozoa, but they are not free. They are not free. They are attached to these Sertoli cells or sustentacular cells. They are attached to the Sertoli cells or sustentacular cells. But in epididymis, they are free. They are free. They are within the lumen. Within the lumen of this duct. Within the lumen of the duct. That is the most important features. That is about the epididymis. Next, coming to the other structure, that is nothing but the continuation of the tail of the epididymis as vas deferens. As vas deferens. This vas deferens, it is a thick, muscular coated tube. And it is having three layers. That is inner longitudinal layer, middle circular layer, and outer longitudinal layer. There are three layers. You see, this is the inner circular longitudinal layer and outer longitudinal layer, and in between these two layers, this is the circular layer. It is having three layers, and the lumen it is a star shaped or it is stellate shaped. Stellate means star. Okay, stellate shaped lumen, and lined by pseudostratified columnar epithelium with the same stereocilia. Pseudostratified columnar epithelium. The epithelium of the epididymis it continues as this vas deferens. Vas deferens. It is also lined by pseudostratified ciliated columnar epithelium with stereocilia. And here within the lumen also there are some amount of spermatozoa. The spans are spermatozoa, mature spermatozoa also present. Okay, this is the very important. This vas deferens, why it is made up of thick muscle wall? Because during ejaculation, it must squeeze or it must contract to push the spermatozoa into the prostatic part of the urethra. For this purpose, it is lined by thick wall. That is the muscles are nothing but the smooth muscles. Muscles are nothing but the smooth muscles. They are arranged in three layers. That is inner longitudinal and outer longitudinal and middle circular layer. This is the circular layer, and this is the outer longitudinal. This is the inner longitudinal, and this is the lumen. The lumen is if you see next is the seminal vesicles. Seminal vesicles, these are also adding bulk to the semen. This bulk to the semen, that is the secretions of the seminal vesicles also adding bulk to the semen. And the seminal vesicles, the most important function of the seminal vesicles is it is providing nutrition to the spermatozoa. It is providing nutrition to the spermatozoa because the secretions of the seminal vesicles, they contain fructose. Ascorbic acid, and prostaglandin. Fructose, ascorbic acid, and prostaglandins. Okay, so these are the structures. They are adding bulk to the semen. At the same time, they are giving nutrition to the spermatozoa because of the content. In the secretions of the seminal vesicles, there are fructose, ascorbic acid, and prostaglandins. These are the ascorbic acid and the prostaglandins are there helpful for the this capacitation of the spermatozoa during fertilization. And the fructose is the main source of nutrition to the spermatozoa during the passage of the spermatozoa from male genital tract to the female genital tract till the fertilization. Okay, that is the most important function of the seminal vesicles. Here, if you want to detect the seminal vesicles, first you have to observe for the mucosa. Mucosa, complicated folds, honeycomb appearance, and pseudostratified low columnar epithelium. There is no cilia. There is no stereocilia here. That is only simple columnar epithelium, and there is a honeycomb appearance because there are so many folds are complicated folds because of presence of the many folds within the lumen of the seminal vesicles. It looks like a honeycomb. That is the characteristic feature of this seminal vesicles mucosa. Seminal vesicles mucosa, and it is lined by pseudostratified low columnar epithelium, and there is no cilia. There is no stereocilia. So that is the point. Next, why there is no cilia? Because there is no propulsion of the spermatozoa. Because the spermatozoa are not at all related with the seminal vesicles. It is only adding bulk to the semen, and also the secretions of the seminal vesicles, they are providing nutrition to the spermatozoa. Because of this reason, there is no cilia. Next, muscle layer. There is a smooth muscle layer. That is muscle fibers are there, inner circular and outer longitudinal. Only there are two layers of the muscles which are present in the wall of the seminal vesicles. These muscles are very important to squeeze the seminal vesicle during ejaculation. Because of this reason, there are two muscles, that is inner circular layer and outer longitudinal. This is the inner circular layer, and here outer longitudinal layer. These are the two layers of muscles which are present in the wall of the seminal vesicles. Seminal vesicles. Next, the adventitia, loose connective tissue containing blood vessels and nerves. Within the outside this smooth muscle layer, there is an adventitia, that is the outer connective tissue layer. It contains blood vessels and nerves. Blood vessels and nerves. Okay, that is the structure of the seminal vesicles. Okay, the most important function of the seminal vesicles is to add the bulk of the semen. At the same time, it also provides nutrition to the spermatozoa. Nutrition to the spermatozoa. That is the most important function. Next, coming to the other important structure, that is in the male reproductive system, that is a prostate gland. Clinically, the prostate gland, it is very, very, very important. In our examination point of view also, prostate gland is very, very important because this is the most commonly affected male gland. Male gland. In the male patients, this is the most common gland enlarged because of the malignancy or because of the benign. It may be benign enlargement or it may be malignant enlargement of the prostate gland. This is the most common gland. It is enlarged and it is troublesome gland to the elderly male patients. Elderly male persons, if there is enlargement of the prostate gland, it may produce obstruction to the urinary flow. Because of this reason, may leads to further, there is always some accumulation of some urine in the bladder and it leads to secondary infections, bacterial infections. Usually what you see, suppose if the water is flowing continuously, it is clear. If there is stagnated water, the algae, fungi or some other insects may grow within the stagnated water. Just like that in our body, if there is an enlargement of the prostate, there is a post-voidal urine. Post-voidal urine. After the urination, going to urination, there is always some amount of urine is retained within the urinary bladder because of the obstruction of the prostate gland. That is the most troublesome part of the male persons after the, especially after the 60 or 65 years of age. Okay, because of this reason, we have to take the biopsy routinely. We are taking the biopsy or we are examining the prostate gland with the by passing the finger through the rectum, rectal examination. That is why the prostate gland is very, very important clinical. Okay, here what are the characteristic features of the prostate gland histology we see. Here, the prostate gland, if you observe the prostate gland slide under the microscope, first you have to observe for the epithelium. That is a prostatic part of the urethra, it is lined by transitional epithelium. Here there is if there is a small movement in the gland, it must be lined by the transitional epithelium or urothelium. Already you know what is the transitional or why it is called transitional epithelium. I don't want to discuss in detail. Okay, next the stroma of the gland is also very, very important. The stroma of the gland is also very, very important. Here the stroma of the gland is fibromuscular stroma. Fibromuscular. This is very, very important characteristic feature of the prostate gland. If you take the breast or mammary gland, female breast, that is fibrofatty stroma. Fibrofatty stroma. Some examiners, external examiners, they may ask, what is the difference between the stroma of the prostate gland and mammary gland? They may ask. Okay, in that occasion, you're supposed to answer. In the prostate gland, there is a fibromuscular stroma. In the mammary gland, there is a fibrofatty stroma. Fibrofatty stroma. Both are glands. They are having glandular tissue. In the mammary gland is also there is a glandular tissue, but may vary. Suppose if the mother is lactating, in the lactating woman, it may glandular tissue is more and fat is less. But if you in the normal breast, the fatty tissue is less, more, and the glandular tissue is less. But that stroma is fibromuscular. This is also very, very important characteristic feature that is of the prostate gland. That is fibromuscular stroma. Fibromuscular stroma. Okay, next, large irregular alveoli lined by simple columnar epithelium. Within the gland, there are alveoli. These are the alveoli. They are responsible for the production of the prostate secretions. Prostatic secretions. These secretions, the prostatic alveoli, they are lined by prostatic alveoli. They are lined by columnar epithelium. Columnar epithelium, that is irregularly alveoli are there, and they are lined by simple columnar epithelium. Simple columnar epithelium. And another characteristic feature of the prostate gland histology is corpora amylacea. They are also called prostatic concretions. Prostatic concretions, they are present in the lumen of the prostatic alveoli. Lumen of the prostatic alveoli. Okay, these are the characteristic features of the histology of the prostate gland. We really observe the back. Okay, see here, this is the prostatic part of the urethra. This is nothing but the urethra, and here it is lined by transitional epithelium. Transitional epithelium, and these are all the glands. These are all the glands. There are three types of glands that are present in the prostate gland. That is smaller and very close to the center of the gland. There are small glands. They are nothing but the mucosal glands. Mucosal glands. And next, outside this mucosal glands, there is some other glands. There are the submucosal glands. Submucosal glands. And finally, the main prostatic glands. Main prostatic glands. There are three types of glands which are present in the prostate gland. That is alveoli. Three types of alveoli. Small segments of the glands. They are secreting the prostatic secretions. Prostatic secretions. This is the lumen, and this is lined by transitional epithelium. These are all the different types of glandular tissue, and in between the glands there is a fibromuscular stroma. This is this is one alveoli. This is another alveoli. In between, this is the fibromuscular stroma. Fibromuscular stroma, and this is the lumen of the alveoli. Within the lumen of the alveoli, there are red bodies. They are nothing but the corpora amylacea. Corpora amylacea. Corpora amylacea. This is the also most important characteristic feature of the prostate gland. If you receive the prostate gland slide, you must write two most important points. One is the presence of the corpora amylacea or prostatic concretions, and number two is fibromuscular stroma. Fibromuscular stroma, and lined by simple columnar epithelium. Alveoli are lined by simple columnar epithelium. These are the very, very important characteristic features of the histology of the prostate gland. Okay, next, because lack of time, I'm going to some fast. Okay, here next coming to the female reproductive system. Female reproductive system. Here the female reproductive system, it contains, already you know, that is a uterus, ovaries, fallopian tubes. The female reproductive system actually it contains internal and external organs. The internal organs are nothing but the uterus, vagina, fallopian tube, ovaries. These are the most important structures which are comes under the internal organs. And external organs already you know, that is a different story. Okay, now we are going to discuss about the female reproductive system histology, especially uterus, ovaries, and fallopian tubes. These are the three most important structures, the internal organs of the female reproductive system. First, we take about the fallopian tube. Fallopian tube or uterine tube. Uterine tube. Here it is the tube. It is responsible for the transportation of the ovum from release of the ovum, that is from the ovary into the cavity of the uterus. Cavity of the uterus. In between the cavity of the uterus and the ovary, there is a connecting tube that is nothing but the fallopian tube. Here, the most important function of the fallopian tube is transportation of the ovum. At the same time, it also provides nutrition to the ovum and fertilized ovum. Or zygote, okay, or up to the stage of blastocyst. Up to the stage of blastocyst is passing through the fallopian tube to perform these two most important functions. That is, number one is the transportation of the ovum, fertilized or non-fertilized. There is different story. Okay, transportation is the most important function. At the same time, it must maintain nutrition to the cells which are passing through the uterine tube. That is, to perform these two functions, be specially designed. The histology of the fallopian tube is specially designed to perform these functions. Here there are highly folded, the mucosa, the fallopian tube presents highly folded mucosa, highly folded mucosa. It is lined by simple columnar epithelium, simple columnar ciliated epithelium, simple columnar ciliated epithelium. The ciliary movements are important to transport the or to move the ovum or zygote from the lumen of the uterine tube towards the cavity of the uterus, and these cilia they move towards the cavity of the uterus towards the center. That is the most important epithelium. That is a ciliated simple columnar epithelium with the cilia. That is the characteristic feature of the epithelium of the uterine tube. At the same time, the mucosa is having so many folds, and it is forming small gaps in between the folds. And these folds and the spaces are very narrow, because these narrow spaces are very important, because of these small narrow spaces, it is able to provide nutrition to the zygote or fertilized ovum in from all sides, because the ovum or zygote, it is large in size when compared with the other cells, other body cells or other cells. The size of the zygote or ovum is big in size, more than 100 microns. Because of this reason, it needs more amount of nutrition to perform this function. The tube of the lumen of the uterine tube, it is divided into small segments, and because of the foldings of the mucosa of the lumen, that is why it looks like a, you see, this is the appearance. It is due to the foldings of the mucosa, and this mucosa it is lined by simple columnar epithelium. That is a ciliated epithelium. That is the number one. Next, behind this columnar epithelium, here there is a lamina propria. This lamina propria also enters into the folds of the lumen. And within the lamina propria, there are blood vessels and nerves, nerves and lymphatics. And with this help of these foldings, it is able to provide nutrition to the zygote or ovum. Okay, when passing through the uterine tube. That is the most important function. Next, outside this, there is a thick inner circular muscle cord and outer longitudinal muscle cord. There are two muscle cords, that is inner circular muscles and outer longitudinal muscles. These two muscles are very important to propel the ovum from the tube towards the cavity of the uterus. To perform this function, it is having two muscle layers, that is the inner circular and outer longitudinal layer. And outside this, there is that is mesothelium, that is nothing but the peritoneal lining. It lines the uterine tube. Okay, that is the important structure of the fallopian tube or uterine tube. Okay, but to perform this function, it is specially designed. Okay, that is the most important of the. Next, outside is lined by mesothelium. This is nothing but the mesothelium. It is nothing but the peritoneum. That is the part of the broad ligament. Okay, that is the fallopian tube. Next, coming to the, you see, they have to draw the fallopian tube. Okay, this is the lumen of the fallopian tube, and this is the wall, and this is the inner circular layer. This is the outer longitudinal layer, and this is the folding, one fold of the mucosa, epithelium mucosa. Into this is the lamina propria. Lamina propria also extends into the folds of the mucosa, and these are the ciliated simple columnar. Okay, that is the most important function. Here, within the lining epithelium of the uterine tube, there are two types of cells. That is, one is the simple columnar cells, simple or ciliated columnar cells. Their function, the function of the ciliated columnar cells is to provide movement to the zygote or fertilized ovum or zygote towards the cavity of the uterus. That is number one. And number two, there are another cells, that is non-ciliated cells. They are also called PEG cells. They are also called PEG cells. They are taller than the columnar cells, simple columnar cells. And the function of these PEG cells are non-ciliated cells is they secrete the mucus. They secrete the mucus. That is the most important function of this PEG cells. PEG cells, the nucleate tube secretions. The secrete tube secretions are from the PEG cells, are non-ciliated cells. That is the important. Then this is the epithelium, that is a muscle layer, that is inner circular layer and outer longitudinal layer. On the outside this is the lining, that is not lining, mesothelium or serosum. This is also called, this is nothing but the peritoneum. This is nothing but the peritoneum. It is having single layer of cells, that is the squamous epithelium. Squamous epithelium. And below this there are blood vessels and nerves, lymphatics. Okay, this is the diagram of the fallopian tube. Next, come to the another important structure, that is uterus. Uterus is the also very important structure in your clinical point of view. Also, the uterus is very important, especially in different diseases or in different clinical conditions. You have to take the histology of the uterus. Histology, you have to observe the histology of the uterus for different purposes. Most commonly, that is infertility, or sometimes there may be diseases, especially carcinoma of the uterus or uterine endometriosis. There are different clinical conditions. Or DU, dysfunctional uterine bleeding. What is the cause? If you want to know, you have to take the biopsy by the process of, usually they do one process, that is D and C. D and C. That is, D means dilatation, C means purities. D and C. This is the most commonly performed procedure in the gynecology practice or obstetrics practice. That is, with the help of this, you can get the this endometrium of the uterus. You can observe the endometrium. Okay, here this is the uterus. It presents what are the important points which are observed in the histology of the uterus. It is already know that uterus, it is having three layers. That is inner endometrium. Outside this endometrium, there is a myometrium. On the outside this myometrium, there is perimetrium. You see, this is nothing but the endometrium. Okay, this layer, this total layer is the endometrium, and outside this is the myometrium. Further outside, you see, this layer is called endometrium, and this total is the myometrium. Then this is the thin layer, outer thin layer is the perimetrium. That is nothing but the peritoneal covering or connective tissue layer. And here this is the uterine endometrium. The uterine endometrium, again it is having two layers. That is the one is a functional layer and one is the basal layer. Okay, you see here is a highly cellular endometrium containing uterine glands. You see here, this is the highly cellular uterine endometrium. These are nothing but the uterine glands. They are nothing but the uterine glands. Okay, they secrete the uterine secretions. Okay, next, it is lined by simple columnar epithelium lining the uterine cavity. It is lined by simple columnar epithelium. Usually, before puberty, it is lined by that is a menarche. Before menarche, it is lined by simple columnar epithelium with cilia. But after the menarche, that is the initiation of menstrual cycle, because of repeated peeling off of the endometrium, there is no development of the cilia. Cilia within the simple columnar epithelium is lining the uterine endometrium. But before puberty, there is a lining epithelium is simple columnar. At the same time, it is also having cilia. That is the most important difference. Next, outside this simple epithelium, and there is a muscle layer, that is a thick myometrium. It is having, again it is divided into three layers. The myometrium of the uterine wall, it is divided into three layers. That is the inner circular layer and middle, that is inner reticular layer, middle circular layer, and outer longitudinal layer. Okay, there are three layers which are present in the uterus, wall of the uterus. You see here, this is the inner circular layer, here reticular layer, and middle circle. Actually, it is not a clear cut. It is not a clear cut demarcation between the layers. All are mixed together, and the circular and the longitudinal also not clearly demarcated. In different areas of the uterus, it is having different structure. Okay, here, suppose if you take this myometrium, it is enormously enlarged during pregnancy. Enormously enlarged during pregnancy because of the hypertrophy and hyperplasia. Hypertrophy and hyperplasia of the smooth muscles of the uterine endometrium. It is a uterine wall, myometrium. It is enormously increased. Almost there is an increase in the size of the uterus 10 times during pregnancy. 10 times there is an increase in the pregnancy, and this increase is possible only with the help of the hyperplasia and hypertrophy of the muscles. Hyperplasia and hypertrophy of the muscles. Okay. Next, we take about the uterine endometrium. Uterine endometrium. Here, this layer is called functional layer. Functional layer, and this layer is called basal layer. Basal layer. Why it is called functional layer? During repeated menstrual cycles, after the menarche, during each menstrual cycle, this layer is peel off and new layer is developed with the help of the basal layer. With the help of the basal layer. And the structure, if you take, that is the thickness of the uterine endometrium is also very, very important. Clinically, normal thickness during proliferative phase, the thickness of the uterine endometrium is 2 to 4 millimeters, 2 to 3 millimeters, 2 to 3 millimeters. But in the secretory phase, that is a phase of ready to receive. That is due to what you call to receive the zygote for the implantation. To receive the zygote for the implantation, at that stage, there is an increase in the thickness of the uterine endometrium. This thickness is increased up to the 4 to 6 millimeters. Maximum is 6 millimeters. Normal thickness of the uterine endometrium is 4 to 6 millimeters. That is after the decidual reaction. Decidual reaction. That is nothing but that is a deposition of lipid droplets, water, and increase in the blood supply to the. Just like if you want to put a seeds in the field, you must give some fertilizer, some water. It must be suitable for the to germinate the seeds. Just like that, here also there are changes which are occurring in the uterine endometrium, especially the functional layer of the uterine endometrium. Normally, the uterine glands are straight, but if you during the secretory phase, they becomes coiled or circular, become coiled or sawtoothed appearance. If you take the section, they may look like a sawtoothed appearance, zigzag. But the basal layer is the blood vessels also same, coiled blood vessels, But in the basal layer, the blood vessels are straight. And the functional layer, the blood vessels are coiled. Okay, because all these changes, they depend upon the hormones of the female. That is especially testosterone, estrogen and progesterone. Estrogen and progesterone. These are the most important hormones. They are responsible for the changes in the uterine endometrium. Changes in the uterine endometrium. That is estrogen and progesterone. Estrogen and progesterone. Because of the effects of these hormones, after the secretory phase, if there is no fertilization, there is a chances for the contraction of the blood vessels which are supplying to the functional layer of the uterine endometrium, and it leads to avascular necrosis of the functional layer of the uterine endometrium, and it may feel off as a menstrual bleeding after that at the end of the 28th day of menstrual cycle. 28th day of menstrual cycle. But the basal layer, there is no change. The basal layer, there is no change. And next, after the proliferative phase, after the menstrual phase, the basal layer proliferates and then it builds the uterine endometrium. That is especially the functional layer. That is the most important histology of the uterine endometrium. Clinically also, the uterus means endometrium. Uterus means endometrium. There are sometimes the thickness of the uterine endometrium will not be sufficient to receive the zygote, receive the blastocyst for the implantation. That is also one of the cause for the female infertility. There is no proper thickening of the uterine endometrium, especially the functioning layer of the uterine endometrium. That is also one of the causes, because there is no proper residual reaction. During that time, there is chances for the abortion, which evacuation of the zygote. Okay, that is the importance of the, or sometimes uterine endometriosis. There are some clinical conditions. That is the importance. Next, the muscle layer. The muscle layer, for theoretical purpose, we study as a inner reticular layer, middle circular layer, and outer longitudinal layer. But actually, there are different direction of muscle fibers. Here, the circular muscle fiber, it is most important. It is more common, more prominent in the cervix of the uterus, because it is responsible for the to retain the contents of the uterus. And muscle longitudinal layer is all over the body. It is more because it is the contraction of longitudinal muscles. It expels the uterine contents. It expels the uterine contents. And the reticular layer, that is also very, very important. This is nothing but the living ligature of the uterus. Living ligature of the uterus, because it prevents the excess blood loss from the uterine cavity. After the parturition, there is a chances for the excess blood loss, and it is prevented with the help of the uterine endometrium, uterine myometrium, that is especially the inner layer, that is a living ligature of the uterus. Living ligature of the uterus. Okay, that is the most important structures that present the histology of the uterus in brief. Okay, next coming to the another important structure, that is ovary. The ovary histology is also very, very important clinically, and routinely we are taking the ovarian biopsy, wherein we are observing the ovaries under the ultrasound scan for different clinical purposes. Okay, here what are the characteristic features of the ovary histology? Growing ovarian follicles at various stages of maturation, that is primordial, primary, secondary, and Broffian follicles. And we are observed. You can observe that if you take the there is adult, there is a female in the reproductive age. Female in the reproductive age, there are different stages of various stages of the development of the or maturation of the ovarian follicles. That is primordial, primary, secondary, and Broffian follicles. Next, the simple cuboidal epithelium. That is nothing but the germinal epithelium, but it is not giving birth to the cells. But it is a misnomer. It is a misnomer. It is wrongly given a name. Germinal epithelium. Outside this one. This is nothing but the germinal epithelium. Germinal epithelium. And but it is not giving birth to any cells. Okay, it is a simple layer, that is a single layer of cells, that is especially the cuboidal cells. Cuboidal cells, they are present in the germinal epithelium. And just below that, this cuboidal epithelium, there is another layer, that is a lamina propria, tunica albuginea. Tunica albuginea. This white layer is nothing but the tunica albuginea, and this dark colored one is the nothing but the germinal epithelium. Germinal epithelium. Below this is the tunica albuginea. And inside this tunica albuginea, there is a structure of the ovary, that is nothing but the cortex. This part of the ovary, we call it as cortex. Outer or peripheral part of the ovary, call it as cortex. And middle part, this part of the ovary, we call it as medulla. This is called medulla. The medulla, it contains blood vessels, nerves, and lymphatics. And periphery, it contains different stages or various stages of the maturing ovum. You see here, these are the primordial germ cells, primordial cells. Okay, primordial follicles. Then this is the secondary, primary follicle, secondary follicle, and this is the Broffian follicle. After the ovulation, and this is the corpus luteum, and then it becomes atretic follicle. White fibrous body, that is nothing but the atretic follicles. And within the medulla, there are blood vessels, nerves, and lymphatics. Here at this side, there are some cells, specialized cells are present. That is, they are called hilar cells. At this area. This is nothing but the hilum of the ovary. That is nothing but the entry point or entry and exit point of the ovary, that is nothing but the hilum. At this area, there are some cells that are called specialized cells, that are hilar cells. Hilar cells, and these cells are probably responsible for the production of the male sex hormone, androgen in the females. Androgen in the females. That is why this is also very, very important. Okay, that is the next here. These are all the different stages of maturation of the ovum. This is the these are all the primordial follicles and primary follicle. This is the primary oocyte. This is the secondary follicle, and this is the Graafian follicle. The Graafian follicle reaches the surface of the ovary. Then it ruptures and it releases the ovum to the surface. And here again, after the rupture and release of the ovum, here then it is converted to corpus luteum. The corpus luteum is also having early stage, that is corpus luteum of menstruation, and then if there is a fertilization occurs, then it becomes corpus luteum of pregnancy. And this corpus luteum, it is also lined by different layers, that is granulosa cells and thecal cells. The granulosa cells and thecal cells are very, very theca interna and theca externa. There are again in thecal cell there are two types of cells: theca interna and theca externa. And theca interna and theca externa cells again they produce hormones, that is testosterone and estrogen and progesterone. These, especially the progesterone, is very, very important for maintenance of the pregnancy. Okay. Then after the, if there is no fertilization occurs, and then it becomes a corpus albicans. Then it becomes a atretic follicle. Okay. That is the, these are all the different stages, and this is the medulla of the ovum. Okay. If you see the diagram, this is the diagram. Okay. This is the diagram of the ovary. There are different stages you have to show. The lining epithelium, the outer lining epithelium, that is nothing but the germinal epithelium. This is simple cuboidal epithelium. Below this is the tunica albuginea. Okay, this is the layer. Then this is the cortex. Within the cortex, there are different stages of maturing. There is a ovum, that is a female gametes. Okay, then this is the Graafian follicle, matured follicle. Finally, it comes to the surface, then it ruptures into the peritoneal cavity. Okay, these are all the different structures which are present in the male and female reproductive system. Okay, thank you.
మరి వాళ్ళకి అవసరం లేదా టెలిమెడిసిన్